Everyone, and welcome to day one of our two-day trainee research presentation series, New Perspectives on Menstrual Health. I'd like to start today with a land acknowledgement. We recognize that we live, work, play, and participate in community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's Call to Action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I am joining you from Toronto, which is covered by the Toronto Purchase Treaty Number 13 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. When we talk about menstrual health, we're talking about something incredibly common, yet still strangely invisible in many parts of research, healthcare, and everyday conversation. And for something that affects so many people across so many years of life, menstrual health is often minimized or treated as something to work around rather than something worth understanding in its own right. Pain is normalized, symptoms are dismissed, and getting clear answers or effective care can take far longer than it should. Menstrual health is not just about cycles. It's a window into broader health and a reflection of how seriously we take the experiences of those affected. When we study it, we are not only filling scientific gaps, we are challenging long-standing assumptions about what gets prioritized in health research and why. So today is about making that invisible space visible. It is about asking better questions, building better evidence, and moving toward care that actually reflects people's lived realities. And so we just really want to thank our trainees for leading this work and to all of you for being part of it. And to start us off today, we have Dr. Mateja Horovic, and I'll have you share your slides.
Can you see that?
Perfect.
Okay, amazing. Hi, everyone. Thank you so much for having me here today. I am a postdoc working with Dr. Lisa Galea at CAMH, but today I will be sharing some of my doctoral research with Dr. Michael Mack at U of T. So I probably don't need to tell this crowd, but we know that ovarian hormones and especially estradiol and progesterone have extensive effects on the brain. One way to study those effects is to leverage the way that ovarian hormones naturally increase and decrease across the human menstrual cycle. So just as a quick recap, in the early follicular phase, levels of ovarian hormones are low. In the late follicular phase, estradiol reaches its peak while progesterone is still low. And over the course of the mid to late luteal phase, progesterone reaches its peak before declining, while estradiol is at a moderate level before it also declines. These changes offer almost natural conditions for researchers interested in teasing apart the effects of different ovarian hormones on the brain. And indeed, there are widespread changes in functional connectivity, brain structure, and task-based activation across the menstrual cycle. In my work, I examine menstrual cycle effects on cognition through a multimodal approach. So using behavioral or task-based measures, neuroimaging, and also considering hormone-gene interactions. Throughout this talk, I will be using category learning as a model of complex cognition. Category learning involves extracting regularities and dissimilarities across experiences to group common elements together into meaningful concepts. This requires careful coordination of learning, memory, and attention, making category learning a good model of complex cognition. Critically, category learning is associated with brain regions known to be affected by hormonal changes across the menstrual cycle, such as hippocampal, prefrontal, and parietal regions. So, to provide a more concrete example, let's say we have the category of birds as animals that have wings and fly. We have to be able to apply those rules to new experiences and recognize, for example, this animal as a bird. However, we also have to be able to detect dissimilarities or exceptional experiences and differentiate them from a given category. So, for example, bats also have wings and they fly, but they are not birds. So, how this looked in the current set of studies is that we showed participants images of flowers and asked them to figure out if a given flower preferred sun or shade. They learned this category structure over 3 learning trials in which they got feedback. And then in the final trial, we tested them without any feedback. Each of these categories, sun and shade, had prototype items here shown in purple. Can everybody see my cursor? Okay, I see Bonnie nodding. Great. So prototype items in purple, these were maximally different across relevant dimensions. Pretty easy to learn. Then each category also had some rule follower items, which looked a lot like their category prototype, also pretty easy to learn. But each category also had an exception item, which actually looked more similar to the opposite category prototype visually. So here you see the solid exception in green, and it's more similar to this dotted prototype of the opposite category than to its own solid prototype. These were very difficult to learn. And the exception items were particularly interesting to us, in part because they are so difficult, and we know that the effects of the menstrual cycle on cognition are quite subtle, and we tend to need more difficult tasks in order to detect them. And additionally, exception learning has been pretty distinctly associated with the hippocampus, which is one of the brain regions most robustly affected by ovarian hormones. So here we have participant performance in that final test block after they've learned the category structure. And we see a distinct effect of the cycle on exception categorization here shown in green. So it starts off in the lower end in the early follicular phase, rises steadily across it, peaks in the late follicular phase, and tapers off across the mid to late luteal phase. We found this pretty exciting because we didn't tell the model where to look for effects, and yet, if you think back to those typical changes in hormone levels across the cycle that I've shown before, this tracks pretty closely with what we would expect to see from estradiol. Another thing to note is that these findings emerged from a novel approach we took to studying the menstrual cycle. You can read a bit more about it in the QR code link that just popped up. But essentially, we treated the cycle as a standardized continuous variable and analyzed it in a nonlinear fashion. So the typical approach in the menstrual cycle literature is to divide the cycle into phases, which are then compared to each other as discrete groups. So if we did that, we would get something like this, where we are still able to say that the late follicular group outperformed the early follicular group, but we would lose out on a lot of this data resolution and all of this within phase variance. So the results of this are discussed in this link, and this one is more so for the methods. But for now, we are able to say that the behavioral effects of the menstrual cycle on category learning are specific to exception items. This is in line with literature showing menstrual cycle effects on cognitive processes necessary for category learning, such as pattern separation, associative memory, and attention. But the findings that I showed on the previous slide were collected online when everything was shut down during COVID, and we couldn't collect any hormone measures. So the next step was to get people to come in person, collect saliva, and run the task in the scanner. Here you first see our behavioral effect. We again see a nonlinear effect specific to exceptions with a mid-cycle peak. For the functional analyses, we first looked at the hippocampus, specifically focusing on subfields along the trisynaptic pathway, which has previously been implicated in exception learning. And we see that ovarian hormones are associated with increased activation for those difficult exception items relative to rule followers. Specifically, progesterone was associated with activation in CA3 and estradiol with activation in CA1, both of which are associated with successful exception learning. Then we took that same standardized nonlinear approach and applied it at the level of each voxel in the brain in order to locate patterns of nonlinear cycle-related activation. We primarily found menstrual cycle effects in prefrontal and parietal regions, which, as you remember, are implicated in category learning. So it's interesting in terms of the task, but it's also cool because it serves as a further validation of this method, and it tells us that we can begin to learn more about brain function by using the menstrual cycle as a continuous variable with nonlinear properties. But things can get just a bit more complicated because we know that ovarian hormones interact with gene expression to predict cognition. A genetic factor of particular interest is brain-derived neurotrophic factor, a protein that affects neuroplasticity throughout the brain, but it is especially highly expressed in the hippocampus. Furthermore, we know that estradiol boosts BDNF expression across the menstrual cycle, and that BDNF genotype interacts with estradiol to predict cognition across the estrous cycle in rodents. So we wondered if BDNF genotype, which we can use to estimate baseline BDNF secretion, would also modulate the menstrual cycle effects on cognition. For this study, we had participants come in twice, once during the early, once during the late follicular phase, trying to get at those low and high estradiol points in the cycle. And again, we see an effect that is specific to those exception items. So people who carry the BDNF met allele outperform those who are homozygous for the VAL allele, but only in the early and not the late follicular phase. And when we look at the underlying hormone effects, we see that in the early follicular phase, when estradiol is lower, MET carriers actually benefit from relatively higher levels of estradiol, but in the late follicular phase, where estradiol is high, this effect reverses. And in fact, the higher the increase in estradiol between the phases, the worse the MET carriers do, and the opposite is true for VAL homozygotes. So, MET carriers may have increased sensitivity to estradiol levels as well as changes in estradiol levels. And this is interesting because we know that people vary in their sensitivity to hormonal changes across the menstrual cycle, especially in terms of affective functioning. There is a lot of work on this in the clinical psychology literature, but here we show a similar effect in a more basic cognitive process and identify a potential genetic factor that can modulate hormone sensitivity. These findings are also interesting because both the menstrual cycle and cognition and the BDNF genotype and cognition literatures are quite rich, but also pretty mixed in terms of findings. So perhaps studying these factors together can help account for some of that variance in results. So in conclusion, I hope I've managed to demonstrate that category learning is a useful model for capturing complex cognition across the menstrual cycle. The set of findings I've shown also has potential clinical implications, as I've touched on in the BDNF slide, and this may particularly be the case for research on psychiatric and neurodegenerative diseases with demonstrated effects of both ovarian hormones and BDNF, such as major depressive disorder or Alzheimer's disease. And finally, on a more methodological note, nonlinear and continuous approaches can provide more nuanced characterization of menstrual cycle effects on brain and behavior. Some specific benefits include increased power and data resolution, reduced bias from individual variance in cycle length, and cycle length is highly variable, and capturing the full extent of variance in outcomes of interest across the cycle instead of binning them into several phase points. And again, I've added the QR code for our methods paper, which validates the method on multimodal data, including basic physiological validation with hormones, the behavioral data that I've shown today, as well as structural and functional MRI data. Thank you all so much for listening. Thanks to my PhD lab and my current lab, and I will now take any questions.
Thank you, Mateus. If anybody has any questions, you can either unmute and ask them yourselves or leave them in the chat. Let's take a look. I can start us off with, oh, Laura has a question. Go ahead, Laura.
Thanks, Bonnie, and thanks, Matteo, for that really beautiful work. Um, yeah, I think this is super important with respect to genetic variability and cycle changes. I know your work is now focusing a bit on menopause, and I was wondering if maybe you could speak to whether you might expect to see similar gene interactions with the menopause transition and cognition, just knowing that in menopause, not everyone shows the same kind of cognitive changes and whether genetic factors might explain that. Thanks so much.
Yes, thanks, Laura, for the kind words and also for the cool question. Yes, I have since shifted to menopause research with Dr. Lisa Galia, and I have actually recently done some analyses that I find pretty exciting with BDNF effects on cognition across the menopausal transition. And this was in longitudinal data looking at MET carriers and people who are homozygous for that VAL allele. And I used the nonlinear model again. And what we see is that while everybody kind of declines in cognitive function, which you would expect to see from aging, the MET carriers actually show an inflection where it looks like cognitive decline accelerates around the time of last menstrual period. So it seems that similarly to what we saw here with the menstrual cycle and hormonal sensitivity, we also see in menopause.
So cool. Thank you.
Thank you.
I guess to piggyback off of Laura's question, I am also really interested in what you think about how aging changes these things. And you touched on other potential individual differences that contribute to variability in the data. So, in addition to aging, you mentioned cycle length variation. So, I'm just curious if you've read more about that and how it might change their ability to perform these cognitive tasks.
Mm. That's a good question. I mostly mentioned the cycle length as a way of boosting that method
Hmm.
comment that I had because you can kind of account for that when you standardize everybody to the same like 0 to 1 scale.
Yep.
Um, that helps put oh my gosh, that helps combine everybody's data more effectively. But it's a really great point. Just generally with menstrual cycle research, usually samples are like normative samples. So there's a cutoff for, you have to have regular
Hmm.
cycles. Your cycle has to be between, 21 and 35 days, which is actually what I did in my work as well. But we might not see similar effects in people with irregular cycles or, for example, in conditions such as PCOS.
Hmm.
So that would definitely be interesting to look at. Also, for example, anovulatory cycles, because that can affect hormone levels throughout the cycle as well. I know that doesn't really answer the question, but we need
No,
to
it's
do more research to
yeah,
figure it out.
yeah, for sure. It's just so many different things that you can look at. I see someone with the raised hand, so if you would like to ask a question, go ahead.
Oh, hello there.
Hello.
I was just curious as to whether there's been research in this area concerning cognitive abilities, concerning women who are either menstruating or on the continuous pill. Yeah, just wondering if you knew anything about that sort of area, please.
Yes, so not with this specific cognitive task. I know that there has been work comparing women on the pill and those who are naturally cycling. It is not fully my area. To my knowledge, it is fairly mixed. And with the hormonal contraceptives, it's difficult to study them because there are different compositions, and it also matters whether you are on the active day of the pill or if it's the placebo period. And also the timing of pill ingestion also matters. I would actually like to plug Laura's work. Laura Gravelsons has a cool review on hormonal contraceptives and cognition in Frontiers in Neuroendocrinology if you want to check that out.
Laura Gravelson was that?
Yes, I
Oh,
can
brilliant.
type her name in the chat after this.
Thank you
I
so
can share
much.
that paper.
So many variables. It would take you a long time to cover them all, wouldn't it? Thank you so much for your insight.
No problem. Um,
Right, perfect. So we can quickly get you to switch slides over, and we'll have Marielle Flores talk next.
I just to confirm that you can see my screen and you can hear me. And nice. So, okay, hi everybody. I'm Mariely Flores Villalobos, a PhD candidate at the School of Public Health at the University of Alberta. The title of my project is Exploring the Time of COVID, Women Menstrual Experiences. So I'll start with the state of connection, then I'll move into the topic, how I'm doing this research, some preliminary highlights. There are not findings because I'm in the middle of the analysis while also doing some focus group, and then we can have a conversation. So first of all, I just want to say thank you to everybody that is here. I know that the world is suffering. There are so many human rights violations happening all over around us, and I'm sure it impacts us in different levels. But thanks for taking the time and being here. So I'm from Peru. My family is from the coast and the highland of Peru. There is a territory ruled years ago, thousands of years ago, by the Moches and the Chimu culture, and then it was conquered by the very famous Incas. I moved to Canada a couple of years ago, actually, yeah, four, um, to do my PhD. And I've been very lucky since the moment I got here to have people around me, like the people in my committee, that has led me actually question who I am in this land and how do I benefit from the ongoing colonization project that targets indigenous people. And I think that is so important for me to say this because I developed this project having that into consideration as well. So what I want to talk about. As you all know, a couple years ago, we had a pandemic, the COVID-19 pandemic. So there are a lot of research about that, but there is also a lot of questions around that. Some of the studies have confirmed that the pandemic context, the COVID-19 virus, and the vaccines, they could cause an impact on your menstrual cycle, resulting in alterations to your sexual and reproductive health. However, they are categorized as abnormalities that will eventually pass, so you don't really have to worry about it. What I'm doing with my research is kind of changing the focus into those research and kind of thinking more on women's experiences. So what I aim to explore is women's relationship to their menstrual cycles to better understand how meaningful it is for them, and with that, analyze if the pandemic has impacted that connection. To do that, however, we need to understand the power structures that we are living in, like biopolitics, neoliberalism, and racialized status, because they can determine the tools that we have to understand what's happening with our own body. And also, we live in a context of systematic violence, erasure, and silence of women's experiences and bodies. Plus, we have a historical exclusion from clinical trials. However, there is a lot of work, as we're seeing right now, of how women are being the focus of this research, which is very important. And I also would like to take some time to acknowledge that gender is a dynamic and performative identification process for the purpose of my research. However, whenever I'm talking about women or people, I'm referring about women who are self-identified as such and who were menstruating during the pandemic. So the big question that guides my research is, what were the experiences of self-identified women who menstruate in Canada and Peru during the COVID-19 pandemic in relation to their menstruation, both when infected with the COVID-19 and after receiving the vaccination? I chose Canada and Peru, not only because I have connections to both of these countries, but also because we're in the Americas, those two countries were the ones with the highest percentage of people vaccinated during the initial protocol with the vaccines. So these questions allow me to focus in three objectives. One is to understand the explanatory models that women use to understand what happens to their bodies. So I'm thinking about culture, religion, spiritual beliefs, education, and with that focus, how our memory and how our body keeps carrying those experiences. And then I also want to really focus on how women take care of themselves to see if the gaps that we have on our public health can be improved somehow with their experiences. How am I doing this? I'm using a qualitative feminist and decolonial methodology. This means that I'm looking to provide a space with myself as a researcher and the participants. We can be very reflect on our power dynamics, but also remain really critical about how we are perceiving our own experiences. I'm using life history interviews, focus group, and art-based methods. Participants are 6 from each country, between 23 and 35 years old. So, highlights. Again, these are not results yet. Um, Again, I've divided them between before, during, and after COVID. So to this to this day, the highlights for me during their experiences of before COVID is that the first time that they got their period, they were between happy because they wanted to belong to this group of women they knew that were bleeding already. Some of them were really scared, didn't know exactly what to do in the moment, and others were really confused and afraid because they didn't understand at all that for them bleeding was a possibility even. Their moms, friends, the school are like scenarios when they learn a little bit more about, oh, this is menstrual cycle and what's the biological meaning of this. And feminism has been a tool for them to understand their body in a wider perspective as well. However, something that is very remarkable is that they found that they need to be a good woman since the moment they got their first period. What does it mean? There's not an answer for them at that moment. And one of the questions that I asked them is, what would you tell your younger self about your period? And the answers are really beautiful and very diverse. The common thread is that they want to tell themselves, the younger self, that it is yours. So it is okay how you experience, it is okay not knowing what to do, it is okay to build a connection with it along your life. During the COVID, there are different experiences here. One of the common ones is that all of them found that they have the opportunity to try new products like the menstrual cup, and they are very fan of it, being at home, having access to water, to a clean washroom, meant for them to have more like advocacy in their own body while bleeding. So menstrual cup was a big deal. Some of them experienced more painful periods or heavy bleeding, while in other cases, they just didn't get their period. And that it was not just something that they took lightly, that's something that really worries them. However, for a few of them, they didn't quite remember exactly how was their period during that time. And they questioned themselves if that was because they normalized already the pain that they had, that they didn't find it like, oh, something different happened in my life during that time. Or it's just like my memory just doesn't remember that time because it was so horrible that I'm just forgetting about it. What they also say is that they found that for the people that did experience the change with their period, that they found that COVID-19, from their perspective, exacerbates previous condition. So when they were teenagers, for some reason, they didn't get the period or they get like very painful period, when they got COVID, they felt that those symptoms appear again, but double or triple. So that was very interesting. And after COVID, what they experience like now is that they have a better understanding of how the period evolves for them. So the connection that they have with the period will change, not just because of COVID, but because of any like key events of their life that had happened, that will make them see their period in a different way. However, they also have this big question about how is the menstrual cycle connected to other health systems within their own bodies, and they want like clear answers about that. Right now, they're open to a friendly connection to the period, whether this means to understand it from a cultural or spiritual understanding, or to feel that it's okay to say I don't have a good relationship with it, and I'm okay to just let it go. And finally, they mentioned how lovely it will be if we understand the bleeding experience as a collective practice, because every human somehow is connected to it, and how painful it is to live your period feeling isolated and alone, and that the social structure where we live, that it is not prepared to support women that are menstruating. And that's it for me. So thank you, and I'm happy to chat.
Thank you. And thank you to everybody who's clapping. It's always really nice to see them on a Zoom call. I have a question to begin with. I really appreciated that you shared a lot of context and kind of where your perspective came from on doing this project. And I really wanted to understand more about your thoughts behind the importance of co-creating content with the communities that you're working with and maybe some of the considerations that you have to take.
Hmm. Thank you for that. Yeah, I believe that um, like I knew since the beginning that I wanted to do my PhD on a topic that can allow me to connect with people.
Mhm.
So I had like an experience that kind of inspired this project. And at the same time, during the pandemic, a lot of people were using social media to share their own experience about bleeding, the questions that arise, and the pandemic itself. So I found that I wanted to keep that spirit through all the project. And I've been very lucky. Like here I've been able like to participate in indigenous ceremony that kind of also value in a different way from when I'm from, how a person can bleed. And I think being open to understand that and understand how colonization in our education system and in our beliefs kind of sometimes I feel block us to understanding a more broader sense what does the blood means for us. I think kind of helped me a lot to understand why I wanted to like have that importance of saying, hey, let's think about this project in a more collective way.
I love that. Thank you for sharing. Does anybody have any other questions? We have maybe enough time for a 30-second question or answer. Okay, here we go.
Hi, thank you for your talk. I had a quick question. I was wondering if you collected on any pieces of information such as age, and you mentioned that people felt more connected to their period and understanding that it's okay for it to change over time. So I was wondering if you noticed any differences in this realization or recognition for maybe women who were younger and have not had as much experience having their period versus women who have had their period for longer.
Okay, thank you. So the people that are participating in this research, they are between 23 and 35 years old. And I haven't noticed like a particular difference or a trend that I could see because it's a small sample as well, right? So it's more a connection between how they are, how do they remember that experience. However, what I could say about that is that what I've seen is a shift for what they express, right? Like before, when they understood what the menstrual cycle, how it should look like, they always wanted to have that goal, right? Like it's supposed to have these days and they later on 7 days of bleeding and that's it. Uh, but as years passed, they realized that their own menstrual cycle didn't fit what they were reading about it. And they shared that with other friends or with their moms, and they realized that, oh, they also have a different experience of their menstrual cycle. And that kind of helped them to say, okay, then it is fine if I don't just follow into this template that I was told. And that gave them some freedom to say, okay, maybe I need to just pay attention how my body shows and how my body connects with this menstrual cycle and how my life is impacting that. Like, so they start to realize that when they're very stressed, it will change. When they're on vacation, it will change. When they through a breakup, it will change. So I think it just gave them a wider understanding of how things will impact their own body. That's what I could say about that.
That's great. Thank you so much.
All right, thank you. So we can now switch to the next set of slides and we'll have Poppy Cooper continuing on the COVID theme for the next talk.
Yes. Can everybody see that and can everyone
Looks
hear
great.
me? Okay, fantastic. All right. Yes, thank you very much for having me. I'm Pop Cooper. I'm a PhD candidate at the London School of Hygiene and Tropical Medicine in the UK. And I'm going to talk about the first study from my PhD, which is the menstrual cycle phase and its association with COVID-19 vaccine outcomes, a study of period tracking app users. So follows on very nicely from the previous talk. So when we set out with this study, we wanted to ask the question, is there an optimal time of the menstrual cycle to receive a vaccine? And by that, we mean, is there a specific point where side effects, which someone might experience from the vaccine, fatigue, headache, pain at the site of injection, would be reduced, and also where perhaps the protective effect of the vaccine might be heightened. And our rationale behind this comes from the well-established observation that women on the whole have a stronger immune response than men, which is a bit of a double-edged sword. So on the one hand, we are slightly better protected from infections. We'll clear pathogens more quickly, but we also, as I'm sure many people here know, are much more susceptible to autoimmune diseases. And in vaccines, that plays out in that we do have a stronger antibody response to a vaccine and tend to get better protection, but we also report side effects much more frequently than men and at a greater severity and number. And the reasons for this, it's not working. There we go. And the reasons for this are partly genetic. So the X chromosome does code for many immune cells. Behavioral women tend to be a little bit more health conscious, more likely to visit the doctor. And the one that we were interested in, which is the hormonal differences. So women will have more estrogen and progesterone and then more testosterone. So when we look at what these hormones do, so on the whole, this is a very complicated area of literature, but on the whole, generally estradiol, which is the estrogen in the menstrual cycle, is immunoenhancing. So can be pro-inflammatory, can increase antibodies, B cell class switching, whereas progesterone tends to be immunosuppressive, and high levels of it during pregnancy can promote an immunotolerant environment for the fetus, and testosterone on the whole is immunosuppressive. So we know that the differences between the sexes in their immune response is largely explained by differences in hormones, but then women's hormones are by no means universal and certainly not static, as Matea very helpfully, thank you very much for doing that for me, explained they fluctuate across the menstrual cycle as well. So I won't repeat what we've been told already, but if we were to look at the menstrual cycle in those two broad phases, estrogen dominates in the follicular phase and then progesterone in the luteal phase. So we might expect a stronger immune response in the follicular phase and a weaker immune response in the luteal phase. But does this affect vaccine outcomes? So again, go back to what we've heard. So there were many reports of women experiencing changes to their menstrual cycles following the COVID-19 vaccinations, longer cycles, heavier flow, more painful, and subsequent research did confirm that there were changes, thankfully largely small and transient. Not to dismiss, I know some women had a very difficult time, but on the whole, these tended to be small and transient. And of particular interest, there were a couple of studies done which looked at the timing of the vaccination. There's one by Alison Edelman and her team in Ohio, and they very interestingly found that of the women who were reporting a change to their cycle, they were largely those vaccinated before ovulation, so in the follicular phase. So there does seem to be a bidirectional relationship there between timing in the cycle and then the impact of the vaccine on the body. But to date, there hadn't been any research on how the cycle phase affects the vaccination outcomes, so the side effects and subsequent infection risk. So that's what we set out to do. We had our two hypotheses. One, individuals vaccinated in the follicular phase will be more likely to report vaccine side effects, more severe ones, and a greater number. And also that those vaccinated in that follicular phase will then have a subsequent lower risk of developing breakthrough infections, which be measured in time to infection. We worked with our industry partner Clue, the period tracking app, and they put out a survey on the app in late 2021, asking app users if they had been vaccinated, the date of their vaccination, which vaccine, and any side effects they had experienced, and then if they had subsequently been infected with COVID. And then also that was matched with the prospectively collected cycle and tracking data from the previous 2 years. And we were able to match over 13,000 of those survey responses to their cycle data. And so we could match the date of their vaccine to the cycle in which they were vaccinated and where in that cycle they were vaccinated. And following exclusions, pregnant women, breastfeeding women, previous COVID infection, anyone on hormonal contraceptives, we had a sample of 1,474, and of those, 760 were vaccinated in the follicular phase and 714 in their luteal phase. And these groups didn't differ in demographic, health, behaviours, vaccination details, or survey timing. So looking through some of the results of our first hypothesis, we did find that those vaccinated in their follicular phase had a 35% higher chance of reporting any side effects than those vaccinated in their luteal phase. And we did various sensitivity analyses here, which largely confirmed our finding, and I just wanted to highlight a couple. So one was we did one sensitivity analysis where we removed anyone who was vaccinated during perimenstruation, so the 2 days prior to the onset of menses and then the first 3 days of the cycle, because they tend to be the symptom heavy days of the cycle. So we were aware that some women may conflate their menstrual symptoms with vaccine symptoms, things like fatigue, headache, which could be either, but we found that the association persisted, in fact slightly strengthened. But what was interesting was when we removed injection site pain, one of the side effects, that association did not quite reach our adjusted significance level, which suggests that perhaps that local side effects may be driving it. When we looked at severity, I just jumped ahead. As Matea pointed out, just looking at the bi-classification of the cycle you can lose a lot of granularity. So we were able to model the cycle on this linear scale from 0 to 1, and look in a bit more granular detail at how those odds of reporting side effects changed across the cycle. So interestingly, here you can see that there is a peak in that early follicular phase, quite likely coinciding with menstruation. Then it does dip and then rises again towards ovulation, possibly as estrogen is rising. And then in the luteal phase is where you have the deepest trough, possibly reflecting that drop in estrogen and the rise in progesterone. And then it rises again towards menstruation, possibly affecting PMT. But as I mentioned, when we did that sensitivity analysis, removing those vaccinated perimenstruation, the difference did persist. But it's nice to see that those two cycle phases are certainly not static or uniform. And when we looked at severity and number, we didn't find that cycle phase associated with either of those. It was just the presence of side effects. And with our second hypothesis, so we looked at the women who had reported that they'd had a COVID infection since the vaccine, and then how long it had been until they had reported that infection, which is particularly relevant for COVID because although the protection was very strong, it did wane over time. So time to infection is an important outcome for measuring vaccine efficacy from the COVID vaccine. And in our crude, unadjusted analysis, we did find that women vaccinated in their follicular phase did report a longer time to infection of about 5 weeks. And you can see that here in the violin plots. You can see the median a bit higher for those vaccinated in their follicular phase. Unfortunately, we did only have 82 infections in our sample, which meant we were underpowered to do a fully adjusted and centered model. So this is very much an exploratory finding and hypothesis generating, which we would like to do more on. So why is this important? Well, there are 1.8 billion women who menstruate monthly around the world, so this affects a lot of people. And these are, as Maria was saying, these women are increasingly educated about their health, about their reproductive health, and are making quite active health decisions. So they also are a group that show greater vaccine hesitancy than other groups, largely because they are concerned about side effects. possibly because this is our theorizing. They're more likely to have children that they're taking care of. Having a day where you feel unwell because of a vaccine costs a lot more to somebody with young children, so they might just say it's not worth it. They're also concerned about the impacts on their reproductive health. And this can have big implications for other people because this is also the demographic who make sure other people are vaccinated. It is mothers who take the children to get their vaccines. It is wives, girlfriends who encourage their partners to get vaccinated. And we see this in Japan, where there was a big anti-HPV vaccine campaign in 2014 to 2019, and many mothers refused to give consent for their daughters to get the HPV vaccine. And as a result of that, it's estimated that there are 10,000 preventable cervical cancer deaths will occur in the next 50 years. So this is an important demographic to build their confidence in vaccines because it not only affects them personally, but other people. So it's important for improving public health outcomes. From our results, as I mentioned, the associations we saw are not driven by perimenstrual symptoms, which suggests that the vaccine outcomes really do differ between the cycle phases. It's possible that this is a result of the immune enhancing effects of estrogen. They do align with what we know about estrogen's effects on inflammation, antibody production, and B cell class switching. Also, fluctuating estrogen levels can modulate pain sensitivity, so it may not be the actual immune system, it may be our perception of how the vaccine is affecting us. But because of various limitations, which we would like to address in further studies, we're not sure. But it's important to acknowledge the data here was self-reported, so there's potential for recall bias. Our app users certainly don't represent all women. The survey was carried out in the US, the UK, Canada, and Australia. It was predominantly a white population, healthy, high income, educated, and health conscious as well. So we want to look at other populations. Ovulation was unconfirmed and estimated. We had to use a calendar-based method. So if cycles were anovulatory, which can affect the hormones, but which would have different hormone levels, we don't know based on the data that we've got. And we also weren't able to take direct immune measurements. We used side effects and time to infection as a proxy. So our results require biological validation. We'd like to do it again with actual hormone measurements, immune markers across different populations and with different vaccines. But for now, I hope this provides proof of concept that menstrual cycle phase should be included as a biological variable in future vaccine research. And if anybody is interested, there is a QR code for the preprint on this. Thank you very much.
Thank you so much. That was really interesting. There is a question from Lisa. And while we address that question, maybe we can get you to switch slides just for the interest of time as well. Lisa, did you want to read your question out loud or did you want me to ask?
I can put my video on, but I look horrid. Nobody cares that I look horrid. Hello, look, I'm so dark. But you mentioned it anyway at the end. I sort of was queuing up that question. But like, are you interested or will you eventually? I know this is an app-based protocol, but looking at inflammatory levels because, you know, rather than thinking, oh, side effects are bad, I don't want to take it because I don't want to feel sick, it actually might indicate greater effectiveness, which is also important for the public health message overall, right? So
yes, absolutely. And yeah, I wasn't able to go into the detail of that, but I think from what I've read of the literature, the sort of the jury's a little bit out on do increased symptoms definitely confer extra protection. They may do, they may not, and certainly be something important to look at. I agree. But yes, I think the benefit of understanding this in more detail just gives women the information so they can make a very informed choice. It may be that you don't feel particularly at risk, but you want to get the vaccine, but you want to minimize side effects. Or maybe you do feel very at risk and therefore you would rather take worse side effects, knowing that it would confer better protection. But it's important that we have all of that information so we can be really clear.
Thank you so much. Okay, so last but not least, we have Beatrice Brundow. And oh, do I see you? Okay, there you go. Sorry, I just wasn't showing my screen. Go ahead. Thank you.
Right, thank you so much. So hi everyone, my name is Beatrice. I'm a fourth year PhD candidate at Rice University in Houston, Texas. And I'm excited to share with y'all today some of the preliminary findings of my study looking at emotion regulation across the menstrual cycle. So I know we already talked a little bit about this, but this always shocked me, and I like to show this for a big picture problem. We have this big blind spot in especially neuroscience research, where within the last 25 years, the number of publications with brain imaging studies showing that any type of women's health related factors included in those studies are very, very limited. In fact, only half a point percentage of brain imaging studies have any specific women's health related physiological measures in this publications. So we have a lot catching up to do. I know it's great to hear that a lot of scientists here are doing this research to push this forward, but for the most part, we've been treating women's physiological measures as noise rather than something meaningful to study. So today we heard a lot about the menstrual cycle, so I won't go too much into it, but for the purpose of my talk, we know that menstrual cycle hormonal fluctuations are very important for brain health because a lot of those receptors for estradiol and progesterone are densely populated in areas like the hippocampus, amygdala, and prefrontal cortex. And those regions are highly involved in emotional processing and emotion regulation. So from a neuroscience perspective, we know that something might be going on there, and the menstrual cycle changes might matter to emotional health. So indeed, there's some research that suggests a certain period of vulnerability within the menstrual cycle. So in the luteal phase, a body of research had found that individuals may experience changes in stress sensitivity, emotional reactivity, and mood. So increased memory for negative stimuli in the luteal phase, as well as increased negative affect symptoms. So with that, I wanted to see how does emotion regulation play into the fact of the changes in menstrual cycle hormone levels as well. And we know that for a subset of group, there is this late luteal affect symptoms that may lead to premenstrual syndrome or PMS. And then in severe cases also leads to a clinical diagnosis of menstrual dysphoric disorders or PMDD. And those two cases are highly correlated with increased emotional reactivity and greater difficulty regulating emotions. But it's important to know that this is not something that everybody experiences. The menstrual cycle and changes in emotions are very, very varied between those that menstruate. So it's important for us to know why that is, why some people feel those symptoms and why others don't, and how can we help those that do. So with that, in our lab, we study emotion regulation through the growth process model. So here's just showing the process of emotion regulation. It has multiple steps, but I'm going to focus here on the evaluation or appraisal step, where we kind of stop to just evaluate the situation that we're in before we feel a full-blown emotion. And specifically in this stage, we can apply something that we call cognitive change or cognitive appraisal, which is just changing the way we think about a situation in order to change how we feel. And this generally in the literature has been associated with better mental health outcomes. And then there's several ways that we can do that within the cognitive change. And one of them that we study is distancing, which is just taking a step back and then viewing whatever you're having, the situation you're having as a third person observer perspective. And then reinterpretation, which is another strategy that we use more commonly on a daily basis, just changing the meaning of a situation, like reappraising or saying, oh, this is going to get better soon. This is usually what we use on a day-to-day. So in previous work in my lab, we found some evidence that emotion regulation might vary across the menstrual cycle. This is a paper that we published last year on Hormones and Behavior Journal. You can scan the QR code to see the full results. So for this study, we looked at emotion regulation and we asked people to regulate with distancing over interpretation. And here you can find emotion regulation scores. So higher means they're able to regulate better. So as you can see, those are in the follicular phase show better regulation with distancing. So they're able to downregulate negative affect more effectively when they're using distancing, particularly in the follicular phase. However, the study had several limitations. This finding we wanted to replicate because, again, it wasn't the main focus of our study, as if you open the study, you see what we focus on was hormonal contraceptives. And also, this is a small sample size, and it was a between subjects design. So we wanted to replicate this result with a more rigorous condition. So that brings me to my current project that I looked at menstrual cycle primarily, and then use the same two strategies to see if they change. And between those two phases, either late follicular or mid luteal. So this is a within subjects design where participants come in twice in the lab during those two phases. And then we check their menstrual cycle with the calendar count and also by taking their salivary estrogen and progesterone assays. So far we're only have females aged 18 to 35, and we assign them to do distancing or interpretation. And the data I'm going to show you today and involves 44 subjects. So we're still in data collection mode. So here's a little snapshot of how the sessions happen. They come into the lab, we take them saliva samples, and then they do the emotion regulation strategy on the computer. So I'm going to zoom in here for you to see how the trial goes. So either they're in the distancing or interpretation group, they're going to see the same thing. So they see the keyword decrease, that's cueing them to regulate their emotion when they see a negative picture on the screen. And then they see a scale where we ask them to rate how they feel from 0 to 100. And then they also see a keyword called look, which is just telling them do not do anything, just respond naturally. And then they see a negative or neutral image, okay? So after that, we take a battery of questionnaires just to get to more of the individual differences of each participant. So here quickly is my simple characteristics. So far, they're very balanced in a match in age and cycle phase. So cycle length is about 29 and average. So again, here we only included people with regular menstrual cycles, which irregular menstrual cycles are another very important area of research, but in this case, we just look at regular. So for my preliminary results, again, this is not the full sample, we're still collecting data, but there's some interesting things showing up already. So for the first two graphs here, you're looking at the graph on the left, and this is showing emotion regulation. So when we ask them to decrease, so more means more negative affect, so lower, less negative affect. And break down by groups. So here we have the group that did distancing, and this is the group that did the interpretation, and this is the follicular and luteal phase. And this graph here, we see the emotion reactivity. So the trials that we asked them only to look and respond naturally to see how they're responding naturally to that picture. So as you can see already, the both of the strategies were effective for them. So they're able to decrease their negative affect, either doing distancing or reinterpretation. So that's great. It means their intervention is working. But as you can see, probably by the lines, there was no effect of cycle phase and also no effect of emotion regulation group. So they're doing just as effective in the follicular and in the luteal phase. And then they're doing just as effective as well with distancing or reinterpretation. So what does that mean? Is the cycle changes does not affect how people regulate, or is there more to the story? So this is the main point that I want to drive in my talk. So the cycle phase here isn't the full story. We have to consider individual differences to get to those emotional changes in the menstrual cycle. And this is exactly what we did. We looked at the premenstrual symptom, and then we saw that the premenstrual symptom were predicting more depressive symptoms for individuals with higher PMS mood symptoms. So again, showing here that the menstrual cycle itself is not a pathology. As we know, we don't want to pathologize it, but for a subset of group of women, they do suffer from those premenstrual symptoms, and that seems to be a point of vulnerability for mental health disorders like depression. And then we also found that those that had higher PMS mood symptoms showed that they use less reappraisal strategies in their daily life. So we saw here a negative correlation between the two, meaning that in their daily life, they might be using strategies that are less helpful to regulate their emotions as they report having more PMS symptoms. So what does that mean? To conclude my talk, so together, this finding suggests that emotional responses in our task seems to be stable across menstrual cycle phases, at least in this sample so far. However, it is very important that we look at individual differences when we look at the menstrual cycle and emotions. And in this case, it seems that premenstrual mood symptoms are more related to how people feel and how they approach regulating their emotions in daily life. And then lastly, I want to really emphasize that when we study mental health and menstrual cycle, it is important that we just not look at phase, because again, the menstrual cycle, it's not a mental health disorder, but it could be a vulnerability for some, and that's why we need to look at those individual differences. So lastly, why does this matter and what's next? even in my work, I have to be very cautious to focus on those individual differences and not just average people out and miss those that might be suffering from some condition related to premenstrual symptoms. So I hope to see more research that does that, and also support more research that looks at women's mental health and hormonal and emotional variability. And finally, to use this knowledge to maybe translate to clinical practice to support those individuals experiencing premenstrual mood symptoms. And with that, I want to thank my incredible labs and my mentors, collaborators. And if you'd like to follow and connect with me, here are my information. Thank you so much.
Thank you so much, Beatrice. So we are right at 2 p.m. So I'm just going to share my closing slide and also give the opportunity for anybody to ask any questions on Zoom if you have a quick one. So I'd just like to thank everybody for sharing such thoughtful, important work today, and to all of you for being part of this conversation. And if today piqued your interest, we hope to see you at our second session next week on May 1st, where we'll continue this discussion on menstrual health. We also invite you to stay connected to the Women's Health Research Cluster. Membership is free, and it's a great way to stay engaged with upcoming events, collaborations, and research in this space. You can scan the QR code here that I put up on the screen if you'd like to join. And we hope to see you again soon.